Hey there. In this video segment on enzymes, I'm going to talk about the types of things that can control how fast enzymes are working. And the fact of the matter is our bodies, most of the reactions are controlled by enzymes, and many of those reactions are a series of many steps. And sometimes we need certain things to be produced at a faster rate, and sometimes we need them to be produced at a slower rate or even stop. So I'm wondering if you can see that on the screen. Might be getting in the way. Our bodies have extremely careful cellular control. And as I had just mentioned, it is very common for biological processes to be multi-step and contain many steps. So sometimes if our bodies need an enzyme to speed up a reaction or start a reaction, it will undergo a process called enzyme activation. And there are lots of mechanisms that can be used, that the body can use for enzyme activation. Any process that slows an enzymatic reaction down or slows down catalysis or stops catalysis is called enzyme inhibition. It's being inhibited, it's being slowed down, it's being maybe even stopped. So two types of enzyme activation and enzyme inhibition that we're going to talk about are feedback control and feed-forward control. And feedback control depends on the product that's being made. And think of an assembly line. We are making lots of product, and all of a sudden we're producing more than what the assembly line can keep up with. So the person at this end of the assembly line is going to send a message down to the beginning of the assembly line, hey, we need to slow things down, or hey, we need to stop this for a bit. So with feedback control, the regulation is by the product. If there's too much product, we need to slow the reaction down or stop it. And what it does is it somehow initiates inhibition or stopping at one of the earlier enzymes in these multi-steps. An example of this is the use of ATP in our body. Our bodies eat food. It breaks down the food to generate energy. And the complexity of the pathways that are involved with that are immense. Ultimately, the outcome is ATP synthesis, and this energy provides the energy to our cells that we need. So sometimes you're just laying around and your body doesn't need a lot of extra energy, so it will be built, the ATP will start building up, and a message will be sent back to one of the earlier enzymes in the sequence, say, hey, slow ATP production down, we don't need as much, so it slows it down or maybe even stops it. So that is an example of a feedback control mechanism. Sometimes the product that's being made can actually bind to, so let's say our product is this circle shape right here. Sometimes it can actually bind to one of these enzymes like this and change the shape of it a little bit. So maybe it has this shape now. So the substrate doesn't fit anymore, it's being blocked. And that is a type of inhibition called non-competitive inhibition, which is going to be discussed a little bit later in this presentation. Sometimes, perhaps the product itself may bind to this spot right here, the active site, and block it from anything entering that active site. and that's called competitive inhibition. It's actually competing for the active site location. And it doesn't always happen this way. Sometimes the excess product will send a chemical signal that somehow initiates inhibition somewhere down the line. So in feedback control, it is ultimately the product that is controlling how the reaction sequence is going. So if we start, if our body starts demanding more ATP, demanding more energy, a signal will be released, and maybe this then leaves, or maybe this leaves, and it opens up that enzyme again to start working its way down the sequence and start making ATP again. So that's just one example, ATP, and there are numerous of them in the body. Feed-forward control activates enzymes to make the process move forward. So with this one, it is not so much the product that's in control, it's the reactant. So we have a lot of reactant and it's building up in excess, so it activates the enzyme, which activates the next, which activates the next, which ends up giving you your product. So it is just a sequential down the line. Feed forward control is when this is in excess, it propels the reaction to make the product. So a closer look at inhibition. An enzyme inhibition can be reversible or irreversible. In the case of ATP here, that's an example of irreversible inhibition, because when we need more ATP, whatever is inhibiting, I think I made a mistake when I said this. This is an example of reversible inhibition, because when we need more ATP, whatever is blocking this enzyme from working can start working again, and we can start making ATP again. So that would be a reversible inhibition. Some inhibitors will remain permanently bound to an enzyme and inhibit it forever. So whether it is reversible inhibition or non-reversible inhibition, there are a couple of ways this can happen, and I mentioned them a little bit earlier. We can have a competitive inhibitor. So this is the inhibitor. It's actually just sitting there right in that site, and this can't get in. Bunk, bunk, bunk. It's not getting in. The thing's right in the way. So if the substrate cannot enter, the reaction cannot proceed. And if it is an irreversible inhibition, it will stay there, it will bind there, it will be stuck. If it's a reversible inhibition, it can leave and the enzyme can continue working. Another way we can get inhibition is called non-competitive inhibition, and there's another name for this. This is also called allosteric. So you may come across that word in some of the videos I've decided to share. The non-competitive inhibitor actually sticks to a different spot on the enzyme, and that triggers some sort of change at the active site to change its shape. So the active site is no longer available. Its shape is not the way it needs to be anymore because this inhibitor is stuck to a different part of the molecule. So here is an application. The question of why heavy metals and pesticides are so dangerous. So these are two examples. Let's talk about these. First, talking about heavy metals. I think I can get rid of this. Oh, there it is. Good. If you couldn't see what I was doing, there's a dock of some sort on the screen and it was blocking my view. I'm hoping that goes away now. All right, mercury and lead and cadmium and arsenic and many heavy metals are considered irreversible inhibitors. And they will specifically bond to the sulfur atoms in cysteine residues. So remember, cysteine amino acid residues can form what are called these disulfide bridges. And here is the 2-cysteine residues. The mercury can bind itself right in that spot. And what happens when it docks there and it permanently attaches is that the active site changes shape. So this is an irreversible inhibitor that is an allosteric type, or the non-competitive type of inhibition, because it's not competing for the active site, it's just attaching to a different spot that changes the shape of the active site. Now, if we have exposure to heavy metals like this, it could eventually eliminate the use of the enzymes in our body, and our body will simply, our systems will stop working, and ultimately can kill us. So another type of irreversible inhibition comes from some insecticides, and this is how it works on the insects. And also dangerous things for humans include nerve gases like sarin and ricin. They're both irreversible inhibitors, and they inhibit a specific enzyme called acetylcholinesterase. So if we're going to activate some acetylcholine and some acetylcholine esterase in our bodies right now. So acetylcholine, and this is how it's spelled, and this is what an abbreviation for it is, makes muscles move. So if you take your hand and you grasp it, the muscles are moving. It's an acetylcholine activation that is making that muscle tense up and contract. When that muscle relaxes, it is this enzyme, acetylcholinesterase, that is acting upon the acetylcholine to break it down and make it go away, so the muscle can relax. If this is blocked in any way, our muscles would tense up and they would just stay that way. So this is this is the acetylcholinesterase enzyme right here, and right here is where the active site is, and here is some sarin gas. And what happens is the sarin gas will chemically bind to that site, so this active site no longer has the same shape. Therefore, that acetylcholinesterase is completely blocked, and it'll actually stop you breathing. So every time you breathe, you inhale and your diaphragm contracts. So the acetylcholine allowed your diaphragm to contract. Now, if you are exposed to nerve gases that block acetylcholinesterase, your acetylcholinesterase cannot work anymore, and you can no longer exhale, and you will ultimately become suffocated. I have another application of acetylcholinesterase, and this is used in medicine. And it's a medicine by the name of succinylcholine, and it is used as a muscle relaxant during minor surgery. So if this is your muscle tissue, and when your muscle tissue contracts, the acetylcholine on there makes it contract. When it relaxes, the acetylcholinesterase gets rid of the acetylcholine to allow the muscle to relax. So acetylcholine actually attaches itself to the muscle, and it forces muscle relaxation. So it triggers the acetylcholine to build up, so that muscle relaxes for a really long time. And then this acetylcholinesterase works on this rather than the acetylcholine, and it as it's working on that, it's not getting to the acetylcholine, and it breaks this down much slower. So that enzyme is acting on this succinylcholine very slowly, compared to how it would break down the acetylcholine so that muscle can stay relaxed for a very, very long time. I believe it is also acetylcholinesterase that is an enzyme that gets inhibited in the production of aspirin, or when aspirin is used in the body, but I can't remember off the top of my head. So some additional discussions on enzyme regulation. There are these things called zymogens. It might be pronounced zymogens. I didn't look it up, I forgot. So some enzymes are synthesized in the body in an inactive form. And activation requires you to cut or split off the molecule. So if this is the zymogen inactive form, what activates it is splitting off the end, and the enzyme then will readjust and make its new shape. So here now we have an active site that wasn't in this spot before. And an example of where this happens in our body is our pancreas makes protease, which is an enzyme that will break down proteins. Now, we don't want this enzyme that's being made in our pancreas to start breaking down the pancreas. That is not good. So what it does is it makes a precursor to protease. It makes a zymogen, and that goes out into the body where it's needed, and it will get cleaved. The enzyme will become active, and it can act, the proteases can start acting on breaking those peptide bonds between the amino acid residues in proteins. Again, our pancreas is made of proteins, so we don't want it working there because it would destroy the proteins in our pancreas. That's not good. So this ends my discussion on enzyme regulation. And as a review, sometimes we want reactions to speed up or start. Sometimes we want them to slow down or stop. If we want it to speed up or start, it's called activation. If we want it to slow down or stop, it's called inhibition. In a feedback control regulation, it is the product that determines how fast or slow the reaction needs to take place. If the product increases to a point where it is in excess, it will trigger some inhibition to make the reaction either slow down or stop. If the product is in need, it will trigger an activation to make the sequence start moving or starting again. Feed forward control is controlled by your starting substance, your reactant, your substrate. If you have that in excess, it starts to trigger that reaction moving forward. Two types of inhibition include reversible and irreversible, and both have many, many examples in science, in our bodies, in nature. And whether it's reversible or irreversible, we can have competitive inhibition in which the inhibitor is taking up the active site, so the substrate cannot enter, and non-competitive in which the inhibitor is binding to a different part of the enzyme, which changes the shape of the active site. And non-competitive inhibition is also known as allosteric control. Heavy metals are irreversible inhibitors that bond in particular to cysteine residues, changes the shape of the enzyme in a non-competitive inhibitor way. And insecticides and nerve gases are competitive inhibitors. The inhibitor actually bonds to the active site, and it is also irreversible. And succinylcholine is used in medicine to slow down the acetylcholinesterase from working on the acetylcholine which allows your muscles to stay relaxed during surgery. And some enzymes we don't want to work where they're made in the body because they could potentially destroy that tissue. And those are called zymogens. They will be activated when a portion of it is cleaved off and the protein or enzyme takes its active shape. So thanks for watching. I am going to talk about some examples of enzymes in medicine and in the real world, in lots of different situations in another segment. So thanks for watching.